Okay. Good morning. Can you hear me? Give me a second. Okay. Can you hear me? You can, right? Yes. Mm. Yanul, Habir, you can hear me, right? Okay. Today we have a very important and special topic. We're going to discuss about microbiology, right? It's a main part in our syllabus. It's a part of cell structure lesson, but it's a main part, right? Our today target complete microorganism and microorganism definition and their characteristic and types of microorganism, different types, different types of microorganism: virus, bacteria, fungi, protozoa, algae. Yeah, mainly four types of bacterial type, I mean, not bacterial, microorganism types. Among them, in industrially for productions, mainly we use bacteria and fungi. Bacteria and fungi mainly used for production process, industrial process. Some protozoa types as well. Okay, one by one we need to discuss their applications and their characteristics and their internal structures and their cell wall structures, nucleus structures, everything. Okay, before moving to our today lesson, today I completely discuss microorganism part, right? Microorganism oil production I will discuss. And evening I start the next part. Yeah, stewed. I got you got all this stewed, right? Kadadunette, around I got. Okay. Okay, no problem. You have soft copy, no? If you don't have microorganism tute, others you all Tanya, you collect. Udanya, Ambunadu. Tanya, yes. Oh, Sarmia. Okay, no problem. It's a very simple part. Microorganism. I will send the soft copy. Then we can start. Give me a second. I will send the soft copy. You can make a short note. Definitely today, if you check the post office, you can collect the tute because last Thursday I posted. Harnang may වැස්සනි සද්දන්නේ. Wait, I will send the soft copy. Then you can start. Okay. I have sent the soft copy. Please check before evening class in your post office, right? You can collect. Hi. Okay. Our today lesson is microorganism. First of all, need to understand about who are the microorganisms. How the microorganism can. Who are the microorganism? Okay, actually, the microorganism, uh, the father or the actual person who found the microorganism is what? What's the name? Who found microorganism? Investigate microorganism initially. Actually, all peoples, ancient peoples, these the ripening of fruits and different uh, diseases especially. Sometimes they think these diseases and some incidents coming from devil and different uh, virtual hazards, right? And they didn't know exact reason for some diseases and some scenarios, right? But there was a scientist, Anton van Leeuwenhoek. He found the, I think in O level time you know this name, Anton van Leeuwenhoek. He found a microscope, very simple microscope combining two lens. He found a simple microscope and he investigate. There are some organisms. There were some organisms we can't see in our naked eye. Some organisms are uh, living around us. We can't see in our naked eye. අපිට මේ naked eye එකට පේන්නේ නැති some organism also there. These organisms are the doing these diseases and some scenarios, right? Ripening of fruits and uh, different diseases. So that, okay. Okay, he is Anton van Leeuwenhoek found a microscope. It's not a light microscope or electronic, very modified microscope. It's very simple type microscope, right? Combining two lens. Okay, actually, he was a scientist. Initially, found there are some organism we can't see in our naked eye, right? Okay, after that there were different types of modified microscope investigate and found different types of microorganism. Not only actually Anton van Leeuwenhoek only introduced there are some organisms we can't see in our naked eye. After when the modification of this microscope, later scientists found these microorganism different types. Then they name as bacteria, fungi, algae, protozoa, and virus as well. Finally found virus. The characteristic is virus, it is very small, and they don't have specific cellular organization. Last day I already explained, right? May virus don't have specific cellular organization. The reason is in atmosphere, in normal environment, these viruses are just dust particle, no any metabolic activities. But when it is penetrate into the cell, when it is penetrate into live cell, it's get nutrition from live cell and it start all metabolic activities. That's the main characteristic of virus. It's very dangerous, right? Normally I told viruses mainly two type, DNA virus and RNA virus. DNA virus mean it's double helix nucleus, double helix DNA. DNA has double helix, then it is stable. This mutation is very low. But if you got COVID-19 type RNA virus, RNA mean single helix. Their genetic material single helix. Single helix. Single helix means it is very unstable. Generation to generation, it's coming mutations. Mutation means they are characteristic. They are this is characteristics. Generation to generation change. That means their strength is generation increasing, right? Generation to generation change due to the unstable genetical material. Unstable genetical material. Hari. Okay, that's a summary. I will start the lesson now. Okay, this is a very, very important lesson and interesting lesson. This is microbiology, right? Yeah, dude. Okay, it's a very long lesson today, right? Very long lesson. Okay. Every primary, very primarily primitive and simple definition. So the microorganism cannot be seen our naked eye. Remember the microorganism full definition. Please note down. Microorganisms are organisms. Microorganisms are organisms less than microorganisms are organisms less than 0.1 millimeter body size. Microorganisms are organisms less than 0.1 millimeter body size and cannot be seen with the naked eye. With the naked eye. Naked eye. Naked eye. Right. This is the microorganism definition. Normally, the microorganisms are the simple definition is microorganisms are organisms we can't be seen our naked eye, right? Remember, normally, think this example. If you got these two dots, if you got two dots like this, right? Now it is two dots. Now it is two dots. Can you see any gap between these two dots in our naked eye? Can you see any gap between these two dots in our naked eye? No gaps, right? It's we can see it's just touchy. But if we zoom this, what happened? There is a very small gap. There is very small gap. Okay, that's why I told you the 0.01. Remember 0.01 millimeter is 0.01 millimeter, not 0.01. 0.1 millimeter is the maximum visibility of our naked eye. Maximum we can separately identify two substance if it is greater than 0.1 or greater than 0.1. If it is less than 0.1 millimeter gap, we can't separately identify. We can't identify separately, right? That's why if it is any substance less than 0.1 millimeter, what's the meaning of 0.1 millimeter? 1 millimeter divided into 10 parts. At the hidden the gap. You know 1 millimeter in your ruler, the smallest gap. If it is divided into 10 parts, that is 0.1 millimeter. If it is less than 0.1 millimeter, if it is less than 0.1 millimeter, you can't separately identify two substance. May gap maker separately identify current, right? The microorganism size, that size, the microorganism also we can't see in our naked eye. Their body size, their cell size is less than 0.1 millimeter. Then microorganisms are organisms. Microorganisms are organisms. They can't see in our naked eye, and their body size, their cellular size is less than 0.1 millimeter. Understood? They are the microorganisms. Right? Tanya? Clear? Don't leave any due to any reason, don't leave anyone until I complete this today lesson. I will completely discuss microorganisms, their characteristic and their productions. Then if you continue this session, end of this session, the microorganism part will be completed. Very simply I will explain every productions and every microorganism characteristics, right? Therefore today session very important. Hurry. Go to lesson. Okay, now if anyone asks who are the microorganisms, how you can explain? Microorganisms are organisms we can't see in our naked eye, and their cellular size is less than 0.1 millimeter. That's why we can't see in our naked eye, right? That's the reason. That's the reason. Microorganisms are organisms we can't see in our naked eye. Why? Their body size, their organism size less than 0.1 millimeter. Less than 0.1 millimeter means their body diameter actually. Think if it is, think their this thickness or diameter 0.01 millimeter. This value we can't see in our naked eye, right? 0.1 millimeter is the maximum our visibility, right? Right. When discussing them, it is essential to have an adequate understanding of their morphological and physiological and molecular biological characteristics. Okay, that's our lesson. We're going to discuss about their morphological and physiological and molecular biology characteristic of different microorganisms. Okay, what do you know? What are the environments? Living microorganism, normally living microorganism, if you got our atmosphere, is it available microorganism or not? Yes or no? In our atmosphere, microorganisms living or not? Yeah, very good. It's a good huge amount. If you got any air sample, it's contain huge amount of microorganism, huge number of microorganism. In normal drinking water, drinking water, yeah, drinking water also. On our skin, on our skin, others, on our skin, is it living microorganism? Dihini. Yeah, inside our digestive system, food digestive system. Yeah, a lot of microorganisms. Inside our respiratory system. Yeah, inside uh, heavy acid. Heavy acid mean HCl, uh, uh, concentrated HCl, concentrated HCl. What do you think? Concentrated HCl. What do you think? Normal pH level 1 to 2 level, heavy acidic solutions. What do you think? Microorganism living or not? Hint. Yes, very good. Yenol. That we called as extreme environments. Normally, this type heavy thermal temperature, that means more than 1800, like heavy 1800 Celsius type heat temperature. අපි කියන ගිනි කඳුවල, right? Fire forms and very acidic conditions, 1 to 2 pH level, very acidic conditions. Normal 13 to 14 type, very basic pH value conditions, right? These environments also microorganisms are living. Okay, there is no ecosystem in biosphere without microorganism. There is no ecosystem in the biosphere without microorganism. Why? For example, on soil, air, water, animals, animal on the animal body and inside the animal body, as well as hot springs. Hot springs mean hot water wells, hot springs with high temperature, and polar regions mean glaciers. Inside glacier also microorganisms living, right? normal microorganisms Normally microorganisms living in normal ecosystem mean normal organisms, animals and plants living environment, microorganism කොහොමත් ඉන්නවා, right? Normal environments. Not only that, microorganisms are living in extreme environment mean plant and animals cannot live, but microorganisms can live in polar regions mean temperature minus values, completely glacier. Inside this glacier also microorganisms living without any problem, right? That's why we call microorganisms has a high concentrated salt and higher acidic environment, and petroleum deposits also inside crude oil drones also contain microorganism. Overpowering large organisms belongs to yeah. That's why, therefore, microorganisms are living in all ecosystems. All ecosystems mean normal environments, normal animals and plants living environments, soil, air, inside and on the animal body. They are normal environment, but not only that, they are living in extreme environment as well. Extreme environments mean hot water stream, very high temperature, right? Very highly concentrated salt, that means seawater, highly acidic conditions, and petroleum deposit, and what else? Inside glacier, microorganisms are living, right? Therefore, as well as normal environment, microorganisms are living in highly extreme environments. ඒ කියන්නේ normal organisms වලට ඉන්න බැරි areas වල පවා microorganisms are living. That's why I told microorganism, there is no ecosystems in biosphere without microbes. Microorganisms are in all ecosystems in the biosphere, right? All ecosystems, not only normal environments. This is example, thermopilus which grow hot springs. Thermopilus types are growing in hot springs, and some pseudomonas types which growing in polar regions. Pseudomonas type in very cold areas. Thermopilus is living in hot springs, very high temperature. Okay, don't misunderstand me balan. මෙන්න මෙහෙමයි වෙන්නේ. Now if you got normal drinking water, හරිද? අපි normal water එකක් ගත්තොත්. Normal water, right? Normal water. It's contain huge number of, more than million number of microorganisms. If it is heated, අපි දැන් මෙයා heat කරොත් මොකද වෙන්නේ? අපි හිතමු 100 celsius වලට heat කරනවා. Heat up to 100 celsius. Then what happened? Lot of microorganisms destroyed. That means some microorganisms destroyed at 40 celsius because their tolerant rate is 40 celsius. Tolerant limit is 40 celsius. Some range 50 celsius. Some range 60 celsius. Some are 60 වෙද්දි මැරෙනවා. හරිද? But some microorganisms are type tolerant up to 70 celsius. But remember, if we heat up to 100 celsius, it's contain some 1% වගේ ඉතුරු වෙනවා. Because they are tolerant to this high temperature. They are thermopilus types. I got you know. ඒ කියන්නේ මේ hot spring වල හැම microorganisms රටම ඉන්න බෑ. Every microorganisms cannot live in hot spring and all extreme environment. They have adapted for environments, these extreme environments. As example, if you the pseudomonas clinic, pseudomonas is adapted for these polar regions, glacier minus temperature. Pseudomonas api mehe gihin dammot, if we add one pseudomonas bacteria into hot spring, can it live? No, it will be destroyed, right? It will be destroyed. Only some microorganisms types adapted to these extreme environments also, right? That's why I told microorganisms can live in normal environment as well as other animals and plants not living extreme environments. Understood? Any doubt? Clear? Hari. Now come to next part. Okay. Now, I told microorganisms living in all ecosystems. No? Other thing, I told microorganisms are living on our skin. Huge number of microorganisms living on our skin, right? It's a good nutrition media for them. That's it. Okay, but remember 0.1 inside our blood, no any microorganisms living inside our blood. That was the MCQ question. Ape blood cell, no any microorganisms living, because we have immune system. Due to the immunity system, if there are any microorganisms enter to our blood, it will be destroyed. This microorganism will be destroyed by our immunity system, right? Ape immunity system making destroyed karna. Therefore, in our blood, healthy person blood, no any microorganism, remember. But in our digestive system, respiratory system, everywhere microorganisms living, but not inside our blood. Okay, not in our blood. not only our blood, any animal blood don't have any microorganism. Okay, the next important part, reasons for ubiquitous behaviors of microorganism. That means I told they can living in extreme environment as well. Reasons for they are living in extreme environment as well. Okay, the first one, they are too small in size. Yeah, the first thing, they are too small in size and they have rapid growing and rapid reproduction rate. Rapid growing, within very some microorganism, few hours they matured and producing new cells. Rapid growth and reproduction rate. Having wide morphological and physiological diversity. Morphological and physiological diversity. And nutritional diversity. Nutritional diversity. Morphological diversity means they have 4 respiratory types. Nutritional diversity means they have another 4 nutritional types. I will explain the next part. Very simple. They were Tamahan exam. Being able to echo, it is producing symbiotic relationship between other organisms like us. Some as example, some probiotic bacteria living in our digestive system. They help to our digestive process and they get nutrition from our foods, right? This probiotic bacteria inside our digestive system, right? They help to our digestive system and they take nutrition by our foods, right? They are the relationship types: symbiotic and parasitic, different relationships, right? Different relationship with microorganisms. Microorganisms between animal and plant, they take their nutrition and keep their living style, right? Okay, what are the characteristics? They have very small cell size and rapid growth and reproduction rate and wide morphological and physiological diversity. That means morphological and physiological diversity mean they have respiratory diversity and nutritional diversity. I will explain later. And it's making relationship between other macroorganisms, symbiotic and parasitic relationships. Symbiotic and parasitic relationship. Hari. It is given, it was mentioned about that organisms that cannot be seen in our naked eye, they are the microorganisms. Object smaller than 0.1 millimeter have been scientifically proven to be undetectable in our naked eye. Yeah, that's I already explained. Microorganisms are organisms less than 0.1 millimeter cell size. Yeah, a light microscope has designed to observe these microorganisms. Lightning designed by Anton van den Hoek. This microscope first investigate and found the Anton van den Hoek. The microscope is progressively improved magnify an object by a 1,000 times and invented microorganism that could observe, right? It's clearly observed by magnify up to 100 times, 1,000 times. Now modern light microscope, we can magnify, we can zoom up to 1,000 times zoom karan pulon. Using improved light microscope, biologists were able to gain some insight into microbiological characteristic of organism. Yeah, not only microorganisms type, we investigating their internal structures as well nowadays due to modern technology, right? Okay, this is a different types of light microscope. Not only that, not only light microscope, nowadays we can use electronic microscope as well. By electronic microscope, we can clearly observe internal organelles of microorganism as well. As example, by light microscope we can only observe bacteria and their movements only, but electronic microscope we can observe their internal organs, their bacterial plasmid, genetic material, ribosomes as well. ඒවත් අපිට observe කරන්න පුළුවන් by electronic microscope, right? Right. Now I told that's the most important part. That's the most important part. දැන් අපි කතා කරපු ඔක්කොම අතන තිබ්බා හරිද උඩ part එකේ. Yeah, everything I given here. Hi. Okay, I gave you 5 main factors. Do you remember Evahano? They always asking. Another, everywhere microorganisms living everywhere in the ecosystem. Everywhere in ecosystem. What are the reasons? They are very small cell size, less than 0.1 millimeter, and they have morphological and morphological diversity. That means respiratory diversity that we can discuss later now. And they have nutritional diversity. Nutritional diversity. What else? They have rapid growing rate and reproduction rate. What else? They have make symbiotic and parasitic relationship between macroorganisms. Anne pahawon. Hari. Revise again. E pahawoni. Monada I can. They are small in size. Microorganism small in size and rapid growing and rapid reproduction rate. Rapid growing and rapid reproduction rate within some microorganism very small time, within few minutes mature and reproducing, right? And they have morphological difference, morphological diversity and nutritional diversity and symbiotic and parasitic relationship with macroorganisms. Remember? Due to these reasons, microorganisms living in everywhere in ecosystem. Matakai? These 5 factors. Okay, 3 factors, 2 factors, but you can remember all factors. Okay. Okay. Jenul? Tanya, remember 5 factors. Okay, again revise. Small in size, less than 0.1 millimeter. Second one, rapid growth and reproduction rate. Third one, morphological diversity. Fourth one, nutritional diversity. Fifth one, parasitic and symbiotic relationship with macroorganisms. I think you can remember. Nitra Mahanavati. Okay, let's move into now. First and second one you can understand, right? They are very small in size, you already know, and rapid growth and reproduction rate, you already know. Let's move into morphological difference. Morphological difference means they have 4 respiratory differences. 4 respiratory types. 4 respiratory types. Mainly metabolic reaction take place due to the respiration, right? Energy for metabolic activity is obtained by the respiration. Hari. Balanda. Microorganisms are classified into 4 main respiratory types. That's why that's the main reason for they can living in extreme environments. That's the main reason. Microorganisms are classified into 4 main respiratory groups. Okay, given the first one, aerobic microorganisms. Aerobic microorganism. The second one, anaerobic microorganisms. Aerobic microorganisms, anaerobic microorganism. The third one important, facultative anaerobic microorganism. Given facultative anaerobic microorganisms. Fourth one, microphilic microorganism. Microphilic microorganism. Microphilic microorganism. Microphilic. Okay, again read these names. Them padam karanda. Don't keep anything after class. Okay, read again. Aerobic microorganisms, anaerobic microorganisms, facultative anaerobic, right? No, aerobic. Facultative anaerobic microorganisms. Next one. Microphilic microorganism. Microphilic microorganism. Give me a second. Okay. Now, what's the meaning of the first one? Aerobic microorganism. Aerobic microorganisms. Aerobic microorganisms. Aerobic microorganism mean they living only. That's a if you don't have to, please make a short note. That's a 4 respiratory type. Taggal diagan nam hadara. 4 respiratory type. Microorganisms mainly 4 respiratory type. We can classify it into 4 respiratory types. What are the types? Aerobic microorganisms, anaerobic microorganisms, facultative anaerobic microorganism, and microaphilic, right? Microphilic क्या क्या microphilic microorganism. microphilic microorganism. Microphilic. The first one, aerobic microorganism. Aerobic microorganism mean aerobic microorganism mean they only living oxygen present environment. Only living in oxygen present environments. Oxygenless environments, oxygenless, that means anaerobic environments, right? Oxygenless environments are toxic. Oxygenless environments toxic, like us. Toxic. Oxygen are doing, yeah, like this. Oxygenless environments are toxic. Example, acetobacter. Acetobacter is a acetobacter that we use for vinegar production. Acetobacter is a type of aerobic microorganism. Acetobacter. Okay, it's only one example. You have a lot of example. I'll give you next in applications. Okay, remember now, aerobic microorganism mean they only living in oxygen present environment. If it is oxygenless environment, it is toxic. Clear? Acetobacter. Acetobacter is example. Acetobacter. Right? Okay, let's move into anaerobic microorganism. Anaerobic microorganisms mean very simple, opposite side. Opposite side mean only living environments without oxygen, without oxygen, without oxygen. Oxygen, oxygen are toxic. Oxygen are toxic. Oxygen tamma meyala toxic. Oxygen toxic, right? Oxygen present environments are toxic, right? Oxygen toxic. Opposite side. Okay, who are the examples? Examples, write down example. Clostridium, Clostridium, Pseudomonas, Pseudomonas, Pseudomonas. Other thing, may Clostridium, Pseudomonas may නම් ලියලා the composite row line, right? And the first letter always capital. You done and not only that, Pseudomonas, Clostridium, methano species used for biogas production. Methano species mean ඒ කියන්නේ ඒක group එකක්, right? Methanococcus, methanobacter ඒ ඔක්කොම anaerobic, right? Understood? Okay. Anaerobic microorganisms, they only living environment without oxygen. Oxygen with environments are toxic. Clear? Sarmia? Environment with oxygens toxic to this microorganism, right? Fatima, clear? Hari. Let's move into next one. This is an important type. Facultative anaerobic. What's the meaning of facultative anaerobic? Facultative anaerobic mean special case, special group. Facultative anaerobic mean එයාලා හරි වැදගත්. හරිද? Facultative anaerobic mean living both environments, both oxygen present and oxygen with or without environment. Without environment. They are living in both environments, no problem. Oxygen, if oxygen present, if oxygen present, doing aerobic respiration, if not oxygen, if not oxygen, doing anaerobic respiration. Okay, that's they have any environment, no problem, right? Anaerobic respiration. The oxygen with environments not toxic. Their name is facultative anaerobic, but oxygen with environment, anaerobic environments are not toxic. Not like anaerobic, right? Anaerobic oxygen environments toxic. Again, normal environment with oxygen, they are toxic. They are marina. But facultative anaerobic, no problem, right? Facultative anaerobic, no problem. Oxygen, if not oxygen present, doing anaerobic respiration. Anaerobic respiration. Okay, who are the examples for facultative anaerobic? Examples for facultative anaerobic. Remember, do you know yeast? Yeast? Do you know yeast? In bakery productions use yeast. Yeast is a fungi. Yeast is a fungi. That's scientific name is Saccharomyces cerevisiae. Saccharomyces cerevisiae. Saccharomyces cerevisiae is a Saccharomyces cerevisiae. Saccharomyces cerevisiae is a facultative anaerobic one. You had draw line, right? Facultative anaerobic. Facultative anaerobic. They are living in both environments, no problem. Avulak nahi. Not like anaerobic. Anaerobic mean they are toxic to aerobic environments, right? But facultative anaerobic mean both environments. Okay, avulak nahi. Right. What's the meaning of microreplic? Microaphilic. Microreplic. Spellings. Wait. Yeah, cerevisiae part wrong. C, no? Yeah, C. Saccharomyces cerevisiae. Okay. Okay. They can live in both environments. They can live in both environments. That's the thing. They can live in both environments. Anaerobic mean they are toxic to oxygen present environment, but facultative anaerobic, not toxic, right? That's the thing. That's the difference. Okay, what's the meaning of microrephilic microorganisms? Microphilic or microaphilic? Microaphilic mean the microair, very small amount of oxygen required. That's the meaning. That's the meaning. Only living 5 to 10%, 5 to 10, very small percent. Normal environment, 21% of oxygen. This normal environment cannot live these microrephilic microorganisms. Only live in environment, very low oxygen level, with 5 to 10% of oxygen concentration environment. 5 to 10% of oxygen level, sorry, 5 to 10% of oxygen present environment. Okay, 5 to 10% of normally inside liquid, inside liquid ඒ කියන්නේ අපි liquid එකක් ඇතුලේ එහෙම ගත්තාම normally low oxygen level, right? Normal this type environment, not atmosphere. අපි මේ වගේ එක just milk sample water bath ඒ වගේ එකක් ගත්තාම yeah, in the open, if it is an open one, in the open surface 20% of oxygen, but in deep layers it's low oxygen level, right? In this area aerophilic microaerophilic microorganisms are living, right? 5 to 10% of oxygens are favorable for 5 to 10% of micro oxygen, very low level of micro oxygen level, right? If it is aerobic environment means 20% oxygen. Anaerobic environment 0% oxygen, both environment toxic, right? Remember, if it is 20% oxygen or 0 oxygen, both environment toxic. The favorable condition 5 to 10% of low level of oxygen level. Understood? Who are the examples? What do you think? Who are the example? Who are the examples? You know, milk curd production use lactobacillus. Lactobacillus species are microaerophilic. Okay, note down. Okay, what we discussed, microorganisms living in all ecosystems. What are the reasons? Small in size, morphological diversity, rapid growing and reproduction rate. And the next one is this is a morphological difference. Morphological mean they have respiration diversity. What are the respiration diversity? 4 respiration types. What are the types? Aerobic, anaerobic, facultative anaerobic, and microaerophilic. right? Microhydrophilic. Clear, sir? Sir, Mia? Understood? These examples you have to remember, right? Microhydrophilic, lactobacillus. Yeast Saccharomyces cerevisiae, facultative anaerobic. Lot of anaerobic microorganisms, Clostridium, Pseudomonas, and Methanol species. Acetobacter is an aerobic microorganism. Acetobacter. Hi. Let's move into nutritional diversity. According to nutritional diversity also, microorganisms we can classified into four main groups. Microorganisms, four main groups according to nutritional diversity. Four categories according to carbon source and it is divided according to carbon source and energy source. Carbon source and energy source, microorganisms we can categorize into four main groups. Little bit, these names little bit difficult, you have to remember. But photoautotrophs. It's very simple, man. Photoautotrophs. The second one, same. Photoheterotrophs. Photoheterotrophs. Third one, chemoautotrophs again. මේ වචන වෙනස්, chemoautotrophs. වචන හතරයි තියෙන්නේ. Chemoheterotrophs. Okay, what are the nutritional diversity? Four nutritional types. What are the types? Photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs. Spell again. Pronounce again. Photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs. Again, photoautotrophs, photoheterotrophs. Very simple. Photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs. Right. Now let's see what are the examples and their characteristics. How they complete their nutritional types. Right. Now remember photoautotrophs. I told this diversity mainly depend on nutritional types. Nutritional types mean carbon and energy source. මම මෙන්න මෙතන ටේබල් එකක් දීලා තියෙනවා මම මේක ෆිල් කරන්න මේකෙන් ඇති. හරිද? මේක ෆිල් කරන්න ඇති උඩේ ඕනේ. හරිද? උඩේ තියෙන ධ්‍යාන තියෙන්නේ මේකම තමයි. Right? The first one, what's the first nutritional type? Photoautotrophs. හරි. Tudes නැති අය මේ ටේබල් එක ගහගන්න. Nutritional types, mainly four types. මෙන්න මෙහෙම එන්නේ. Just only table you need to remember, right? Table is enough. First one, what's the first energy source? Not energy source, nutritional type. Photoautotroph. 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 Second one, photoheterotrophs. Photoheterotrophs. Third one, chemoautotrophs. Final one, chemoheterotrophs. Let's see, heterotrophs. Right? Matakai? Sharon? Do you remember four names? Photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs. Udanya? Remember? Fatima? Kabir? Photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs. Tanya, clear? Hi. Now, it is very simple. Now, first one, what are the energy source, what is the carbon source, and what are the examples? That enough. Now, if it is autotrophs, remember the energy source. Photo mean, what is the meaning of photo? Photo mean light. Sunlight is the energy source. Sunlight. Sunlight. Sunlight is energy source. Light. Light energy is the energy source. Then what is a carbon source? Autotrophs. Autotrophs mean they producing foods, their own foods. Therefore, for photosynthesis, carbon dioxide is a carbon source. Carbon dioxide is a carbon source. What are the examples? Remember the examples. The first one, cyanobacteria. Cyanobacteria. It is first one capital, cyanobacteria. Cyanobacteria. Cyanobacteria, there is another name, green sulfur bacteria. Green sulfur bacteria also cyanobacteria, right? Green sulfur bacteria. මෙතන ඒකක් වෙනවා. Cyanobacteria. Cyanobacteria. This is spellings not asking me. Spellings බලන්න නෑ ඔයාලා. හරිද? මේකේ ඒකක් එහෙම අඩු වුණා කියලා no marks reductions. Cyanobacteria. Cyanobacteria. There is another name, green sulfur bacteria. 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 Okay, that's also cyanobacterial type, right? Green sulfur bacteria also cyanobacterial type. Hari. Come to second one. Then මොනවද මතක තියාගන්න ඕනේ? Autotrophs mean they are energy source sunlight, and due to their autotrophs, photo mean sunlight, autotrophs mean they doing photosynthesis, therefore carbon dioxide is a carbon source. මතකයි? හරි. Next one. Second one, photoheterotrophs. Photo තියෙනවනේ. එහෙනම් sunlight. Sunlight. Sunlight is energy source, but it is heterotrophs, no photosynthesis. That depend on another source. No photosynthesis. Therefore, what is a carbon source? Do you know? What's a carbon source? Very good. Organic compounds. Organic compounds mean organic matter. Organic compounds that depend on another substance. They're not producing photosynthesis. They're not producing foods. Organic compounds. Organic compounds. What are the example? What are the example? What are the example? Greens are for drops. Remember that non-green sulfur bacteria. Non-green sulfur bacteria. Non-green sulfur bacteria. Non-green sulfur bacteria. Again, the opposite, right? Non-green sulfur bacteria. Green sulfur bacteria, cyanobacteria, but it is not photosynthesis. Non-green sulfur bacteria. Non-green sulfur bacteria. Non-green sulfur bacteria. Non-green sulfur bacteria. Right. Let's move into chemoautotropes. Chemoautotropes. Hurry. Under chemo. Then what is the energy source? Is it sunlight? No, no, no. Chemo means chemicals. Chemo means chemicals. Then what is the energy source? Do you have any idea? Chemicals. Tama use karan. Any idea? Chemoautotropes. Remember, it is chemicals, no? Chemicals, inorganic compounds. Inorganic compounds. Inorganic compounds. Inorganic compounds. Inorganic compounds. Inorganic compounds. If it is inorganic compounds, then what is it? It is chemoautotropes. Autotropes mean carbon dioxide. Carbon dioxide. Autotropes mean carbon dioxide. Remember, it has specific equations may examples. The first one, nitrosomonas. 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 Nitrobacter. Nitrobacter. Nitrosomonas, nitrobacter are the example. ඔය syllabus එකේ දීලා තියෙන ඒවමයි. හරි. They are the examples given in the syllabus. Remember examples. Nitrosomonas, nitrobacter, both are nitrification bacteria. They come in nitrification, right? I will explain what is the nitrification. Chemoheterotropic. Chemoheterotropic, final one. Normally animals api chemoheterotropic. Then what is our energy source? Api kohomada energy ganna? From sunlight. Sharon, how we get the energy? You know, how we get the energy? Idea? Tanya, how we get the energy? By foods, no? Api sunlight වලින් ගන්න by foods. Yes, then the energy source organic compounds. Api organic compounds sama foods විදියට ගන්න. Organic compounds. What is our carbon source? Api කොහෙන්ද carbon ගන්න? Carbon also, not carbon dioxide, organic compounds. Organic compounds. Organic compounds. Hurry, complete the table. Then what are the example? What are the example? One of the examples. First one, animals. We discussing about me microorganisms. එතකොට animals ඕනේ. Fungi, protozoa, lot of bacteria. That mean අරවට අයිති වෙන්නේ නැති bacteria. Lot of bacteria. Lot of bacteria. This is the summary. This is a summary. This is a summary. We have four nutritional types. Photoautotropes, photoheterotrophs, chemoautotropes, and chemoheterotrophs. Photoautotropes mean they're doing photosynthesis using sunlight and carbon dioxide. Cyanobacteria are examples. Photoheterotropics mean they're using sunlight but not doing photosynthesis. Then organic compounds, they depend on organic compounds, non-green sulfur bacteria. Chemoautotropes mean they depend on inorganic compounds for chemicals for energy source, and carbon dioxide use because they're producing foods using chemicals, right? Nitrosomonas and nitrobacter are example. Chemoheterotrophs, organic compounds used for both energy and carbon source. Fungi, protozoa, and lot of bacteria example. Any doubt you can ask now. Hari? Dihini understood? Nutritional diversity? Dharmya? Give me a second. Okay. Hi. The first part is done. The lesson first part is done. Okay, I will summarize what we discussed up to now. Okay, first we discussed who are the microorganisms. Microorganisms are organisms less than 0.1 millimeter body size and cannot see in our naked eye. Microorganisms living in everywhere in ecosystem, even normally normal environment as well as extreme environments. Normal organisms not living extreme environments. What are the reasons for microorganisms living everywhere? They have small body size, morphological diversity, high reproduction and growth rate, and respiratory diversity, nutritional diversity, and symbiotic and parasitic relationship with macroorganisms. Madakai? What are the nutritional diversity? Nutritional diversity 4 types. Aerobic, anaerobic, facultative anaerobic, and microaerophilic. 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 First one, aerobic mean they only live in aerobic environment, oxygen with environments. Example, Acetobacter. Anaerobic mean they only live in oxygen absent environment, without oxygen environment. Examples, Methanospecious, Clostridium pseudomonas. Facultative anaerobic, only living in, not only, both okay. Facultative anaerobic mean both okay, oxygen with or without, no problem. What are the example? Saccharomyces, yeast. Final one, microaerophilic. Microaerophilic mean it is 5 to 10 very low oxygen levels, living low oxygen levels. Examples, lactobacillus. Okay, right? Nutritional diversity. According to nutritional types, there are 4 main types according to carbon source and energy source. The first, photoautotrophs, photoheterotrophs, chemoautotrophs, chemoheterotrophs. Photoautotrophs, sunlight and carbon dioxide, they doing photosynthesis, cyanobacteria example. Photoheterotrophs, photo mean energy source sunlight and carbon organic compounds used as carbon source. Non-green sulfur bacteria example. Chemoautotrophs, chemicals, inorganic compounds used for energy source, carbon dioxide, autotropic. Since they are autotropics, carbon dioxide used as a carbon source. Nitrosomonas, Nitrobacter example. Chemoheterotropic mean like animals, they use organic compounds for both energy and carbon source. Example, fungi, protozoa, and other bacteria. Methanol other bacteria. I'm not adding animals. Animals also, I mean, plants, plants, autotrophs. Plants we discussing microorganisms, animals into this group, right? First part is done. First part is done. Hari Matakai, Sharo. Don't keep anything to do later. Right? Damn Ivarakara. Tanya, Matakai, remember? Fatima, remember? Abir? Alright, let's move into production process. The production basic part is completed. Let's move into 4.3, third part of the lesson. The first part we already discussed last day. The second part is done now. Third part, the utilization of microorganism for technological applications, technological productions. Okay, the first thing, why microorganisms involved for the production process? What are the advantage? Normally, chemical industries, we already discussed soap production, biodiesel production, biodiesel production, paint production, and phosphate fertilizer production, we already discussed. But what is the main problem in chemical productions? Chemical production main problem is it need huge electricity. It need huge energy cost. But if you use microorganisms already gain sunlight and they're producing their own energy. Therefore, we don't need to provide electricity, right? That is the advantage. Not only that, if you use chemicals, it's producing a lot of byproducts. No, that's a raw material wastage. But if you use microorganism, it is very low byproduct production. Sometimes these byproducts we have a lot of valuable, we can use as valuable product. Not only that, microorganisms living in extreme environments. As example, methanobacter type microorganism, they are living in extreme anaerobic conditions, and they producing valuable energy as well. Energy use energy that we call as biogas, right? Therefore, lot of advantage of using microorganism in industrial process. Understood? Industrial process. But contribution of microorganisms to agriculture. Agriculture. Okay, it's a very important part. Actually, this part is most important. More than previous part. Don't keep anything to do later. Right? That this is the time. You don't have more time. You have 40 days again. Right? The mail is the cell structure. We have evening time as well. Then next tomorrow we can complete tomorrow, and we have Friday 2 hours of the Kanpulan. Then environment chemistry, then the syllabus is complete, right? This is a final week. Okay, next month we can fully discuss papers. I have make a special schedule for final month. We can discuss papers. Okay. Okay, the first one, contribution of microorganisms to agriculture. How we use microorganism for agriculture? You know, the agriculture mainly depend on soil fertility, right? Soil fertility. That means the mainly the agricultural harvest is depend on the soil fertility. If it is low fertility, low harvest. If it is well fertilized soil, fertility soil, we can get large harvest. Large harvest. Large harvest. Okay, to keep the these mineral nutrients are supplied to the soil by decomposition of dead plant and animal parts in the digestive rocks. Right? A dead plant and animal part digestion by microorganisms are mixing with soil. That's why the nutrition level of the soil fertility is made. Soil act as a suitable medium for growth of microorganisms both physically and chemically. Complex organic compounds are simplified by maintaining optimal microbial activity. Yeah, in the optimum microbial conditions, these complex organic compounds like cellulose, it's break down into carbon, hydrogen, oxygen. They break down, hydrolysis process. For this breakdown and keep the soil fertility, microorganisms are they're doing a very important process. Very important process. Okay, to keep the soil fertility, it is very important to maintain environment balance. That means normally, as example, if you got nitrogen cycle, now in our atmosphere 78% of 71% of or 78% of nitrogen. Yeah, 78% of nitrogen in our atmosphere, but in our soil plant cannot absorb as nitrogen gas, need to convert into nitrate. To keep this balance, nitrate and nitrogen, it's mainly contribute microorganisms. The contribution of microorganism as decomposition and nitrogen screen in maintaining the carbon cycle and nitrogen cycle essential to agriculture sector to keep the soil fertility. The maintain cycles in balance stage, it is very important microorganism activity. It is very important to keep soil fertility for agriculture. Right? Api balamu nitrogen cycle හැමදාම අහන ටික. හැමදාම අහනවා nitrogen cycle ගැන. හරි. බලන්න. Okay, it is very important. In this lesson, it's only given microorganism involvement. If you got environment chemistry, අපි අනිත්‍යුත් එක්ක ගත්තොත් ඒකෙත් තියෙනවා that one also given nitrogen cycle. The cyclic of matter, the same thing we discussing, right? හරි මම මෙතනදී කියලා දෙන්නම් full nitrogen cycle, then it is common for both lessons. හරි nitrogen cycle කියලා එකයි තියෙන්නේ මේ lessons දෙකේ වෙන වෙනම දීලා තිබ්බට. Right. Yeah, then it is easy for next lesson. හරි බලන්න. You know, nitrogen is a major component. You know, main nutrition. If you got nutrition to develop plant, mainly required main nutrition, plant nutritions we can classify it into BST students already know. Plant nutritions we can classify it into two major groups: macronutrients and micronutrients. Macronutrients and micronutrients are nutrients required in large quantities. Godak koneva. Monada Tanya, what are the macronutrients? Macro plant nutritions. Udarne. Yeah, macro plants nutritions: carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium. Mainly carbon, hydrogen, oxygen. Carbon provide from atmosphere. Carbon dioxide with it absorb karna. Hydrogen and oxygen provide by water. Then carbon dioxide and water provide carbon, hydrogen, oxygen. Therefore, no limitation carbon, hydrogen, and oxygen. After carbon, hydrogen, and oxygen, the main nutrition is nitrogen. The second one is phosphorus. We use apatite phosphate, triple superphosphate. The third one is potassium. NPK. Remember, main plant nutritions. NPK. NPK. Right? The nitrogen is a main requirement. Nitrogen is a main plant macronutrition. Macronutrition requirement. Macronutrition requirement. හරිද? මේ බලන්න. Normally in our atmosphere 78% large percentage of nitrogen gas, but like carbon dioxide, nitrogen cannot absorb as nitrogen gas. Nitrogen gas විදියට absorb වෙන්න බෑ. Right? Plants cannot absorb nitrogen as nitrogen gas. Carbon dioxide absorbed for photosynthesis process, but nitrogen is not absorbing as nitrogen gas. Right? Give me a second. Okay, what I told you, the main macronutrition for plant growth, nitrogen, but 78% nitrogen in the atmosphere, 78% nitrogen in the atmosphere, but plant cannot absorb as nitrogen gas, right? That's the problem. That's the problem. If it is absorbed, no main problem actually. We have a lot of harvest. If it is 98, enough amount of nitrogen, but cannot absorb as nitrogen gas because it is very unstable, right? And no option to plants. Carbon dioxide absorb for photosynthesis process, but no nitrogen absorption method. Okay, therefore, if we need to absorb nitrogen, nitrogen absorption, this nitrogen need to be converted into nitrogen gas, need to be converted into NO3 nitrate ion. මෙයාගේ නම මොකද්ද? nitrate ion. Nitrate ion. Nitrogen gas need to be converted into nitrate ion. This nitrate ion dissolved in water, then water absorb කරනවත් එක්කම as a mineral, as an ion, this nitrate also absorbs to plant. This is the main method, absorb nitrogen to plant, right? Through the root system. After dissolving nitrate ion in the water. Understood? That's conversion process. Nitrogen gas conversion into nitrate, this process we called as nitrification. 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 This nitrogen gas convert into nitrate ions that we called as nitrification. There is another process. This after when it is converted into nitrate ion again convert into nitrogen gas in anaerobic conditions. This nitrification is take place in aerobic condition with oxygen. Oxygen tibbot tama nitrification වෙන්නේ because it's doing by aerobic microorganism. I'll explain who are the microorganism. Aerobic microorganism. Nitrosomonas, nitrobacter tama involved වෙන්නේ mainly. Not only that, rhizobium, තව ගොඩක් අය ඉන්නවා, anabina, clostridium, ගොඩක් අය ඉන්නවා. I'll explain one by one. However, it is convert nitrogen into nitrate in aerobic condition. It's an aerobic process. Aerobic process. It should have oxygen, right? Nitrate. Remember, if it is anaerobic condition, anaerobic condition එකක් වෙන්නේ කොහොමද? Then think in flooding season, flood වුණාන් පස්සේ all pores in the soil completely closed by water, completely filled by water. Then no gases mean it's anaerobic conditions. Anaerobic condition වලදී nitrate මොකද වෙන්නේ? nitrogen වෙනවා. That we called as denitrification. Denitrification. Normally 78% of nitrogen in atmosphere, but plant cannot absorb as nitrogen gas. Therefore, nitrogen gas using microorganism, it's converted into nitrate ions. This nitrate ion dissolved in water and absorbed to plant. That's the main nutrition supply method to plant. It takes place in aerobic microorganism under aerobic conditions. But if it is completely soil closed by water, filled by water, that means no gases. It is anaerobic condition. In anaerobic conditions, soil nitrate ions, dissolved nitrate ion directly convert into nitrogen gas by anaerobic microorganism. Up to now, okay? Nitrification and denitrification. Nitrification and denitrification. Okay. Janul? Sharon? Then make a 100 balance. That's a full nitrogen cycle, full explanation. Hari, 100 balance. If you want short note, please note down, right? I'm not giving time. You have note down, right? This is full nitrification. Okay, now what is the meaning of actually the nitrogen cycle? Mean nitrification and denitrification. In aerobic condition, nitrogen converted nitrate, anaerobic condition, nitrate converted nitrogen. It's a balanced system. That's why the nitrogen cycle is flowing, right? Nitrogen into nitrate, nitrate into nitrogen, nitrate into nitrogen, nitrogen into nitrate. That's a balanced system, right? That's a nitrogen cycle. That's the nitrogen cycle. Okay. In nitrogen cycle, that's the first step. That's the first step. හොඳට බලන්න. First of all, nitrogen gas, it's convert into nitrogen gas convert into NH4 plus ion. What is the name of this ion? NH4 plus ion. NH4 plus ion. Tanya, what is the name of this ion? NH4 plus ion. NH4 plus ion. NH4 plus ion. NH4 plus ion. What's the name of this ion? That is ammonium. Very important. Ammonium. 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 Right? Ammonium plus ion. Ammonium plus ion. In the first step of nitrogen cycle, nitrogen gas in the atmosphere convert into ammonium ion. Ammonium ion. There are four main methods of this that we call as ammonification. 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 Yes. Ammonification. 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 Nitrogen to ammonium. Nitrogen to ammonium. Or this process we called as nitrogen fixing process or nitrogen fixation process. Nitrogen fixation process. However, what happened in nitrogen fixation? Nitrogen gas convert into ammonia. Not ammonia, ammonium. Ammonia is ammonia mean NO3, it's a gas. Ammonia is a gas, NO3. Sorry, NH3. Ammonia. Ammonia mean NH4, it's a ion. Ammonium is a plus ion. Hari? Matakai? Salmia? Ammonia and ammonia. In ammonification process, it's producing ammonium ion. Ammonium ion. Right? Hari. Then balanda. This nitrogen fixing process, there are four main types. Balanda. The first one, what happened? Proteins. The first one. Hari. First step. Box brackets. This is the first step. First step. First step. First subtopic. Sub step. Right? Eka vena vidhi. First nitrogen nitrate vena vidhi. Right? Types of nitrogen converted to ammonium. Nitrogen converted to ammonium. That's the first step. Eka vena sub step sama meva. Harida. E vena eka vidhi. Okay. First one. What happened in main? Dead plant and animal material. Dead plant and animal material contain proteins, right? Contain proteins. Dead plant and animal material contain proteins. These proteins digest by microorganism. Digest. Maybe microorganism, right? Different microorganism involved for this digestion process. Normally break down peptide bonds. Protein digest. Then it is produced what? Protein digest kara මොකද දන්න මොනවද හැදෙන්නේ? Tanya, Dihini, in the digestion of protein මොනවද හැදෙන්නේ? They convert into their building blocks. What are the building blocks of protein? We don't have time. Protein digest and break down their peptide bonds. What is produced? Tanya. Very good, Udanya. yeah, before these molecules ever break down වෙනන්නේ උදන්න amino acid වලට නේද? The proteins made by amino acid නේ. Yeah, good, you are correct. But ඊට කලින් it's converted to amino acid නේ. Amino acid. This amino acid contain nitrogen. This is amino acid, I think you can remember, COOH, hydrogen, NH2 amine group. This amine group contain nitrogen, right? It's contain carbon, hydrogen, oxygen, nitrogen. Main nitrogen combine with hydrogen, it's convert into, again using to microorganisms, it's again convert into NH4. 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 This is a one ammonification step, one ammonification step, one ammonification type, not a step. ඒක එක විදියක්. හරිද? One type of ammonification, protein digest to amino acid, amino acid converted, amino acid contain nitrogen, nitrogen contain amino acid converted into ammonia. Ammonium ion, sorry, not ammonia. Have the first step okay? First type okay? First type okay? First type okay? Quickly, quickly. I need response. Okay. Second one, biological fixation. Biological fixation. Biological fixation. Biological fixation. Make a biological fixation type එකක් තමයි, but it is protein digestion fixation. Biological fixation. In the biological fixation process, some microorganisms are symbiotic relationship with plants. Example, symbiotic can ඒවා plant එක helpful. ඒ කියන්නේ එකට ඉඳලා nitrogen fix කරනවා, right? The first one, symbiotic. Symbiotic. And there are another microorganisms, they freely available in the soil. They freely available and doing this nitrogen fixation process. Freely available and doing nitrogen fixation process. Again, නිකන් soil එකේ ඉන්නවා. Between soil particles and they involve for nitrogen fixation process. Okay? හරි. මෙතන අපි ඔයාලගේ names ටිකක් දාගමු. මේ symbiotic example, rhizobium, you know that they are living in leguminous plant root nodules, sorry. Again, leguminous plants are done. Do you know leguminous plants? Leguminous plants mean soybean. Yeah, soybean type leguminous plants, they have root nodules. අපි කියන මූල ගැටිති කියලා. Root nodules. These root nodules contain this rhizobium bacteria. This rhizobium bacteria, what they doing? They absorb nitrogen. They utilize nitrogen as nitrate. As a result of using nitrogen, these bacteria, the plant obtain nitrate. Plant එකට හම්බ වෙනවා nitrate ions. Sorry, ammonia. Not nitrate, ammonium ion. Understood? That we call as symbiotic relationship, symbiotic nitrogen fixation. Clear? With the help of plant, you can plant එකක ඉඳගෙන nitrogen fixing කරනවා. Right? Clear? Tanya? Symbiotic relationship with the help of plant. Right? Rhizobium example. When mom cute take Yeah. Rhizobium, remember this example, symbiotic relationship making rhizobium and anabina. I forgot this name. Rhizobium and anabina. Rhizobium and anabina. Rhizobium and rhizobium. Rhizobium and anabina. What are the names? Rhizobium and anabina. Anabina, right? Freely available microorganism in the soil doing nitrogen fixation process, acetobacter. Don't mix it acetobacter. Acetobacter involved for vinegar production. Acetobacter. 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 Clostridium. 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 Acetobacter. Clostridium. Cyanobacteria. That means green sulfur bacteria. Cyanobacteria. Cyanobacteria. Hari. Onnavatama freely available microorganisms types. Freely available microorganisms type. You have remember. Hari. Clear? Kivan symbiotic relationship produced with plants, rhizobium and anabina. Freely available Azotobacter, Clostridium and Cyanobacteria. Hari. Now what they're doing, they convert nitrogen into, they produce nitrogen into NH4. ඔන්න යාලත් හදනවා nitrogen, sorry, ammonium ions. Okay, okay, okay. Okay. Dihini, clear? Hari. Now this is a microorganism involvement, not only microorganism. දැන් අපිට ඊළඟ lesson එකේ මෙහෙම තියෙනවා. For this මේක විතරයි වැඩි වෙන්නේ ඊළඟ lesson එකේ. හරිද? For this nitrogen fixation process මේ වැඩේට there are two another process involving that we called as the third step, third process, atmospheric nitrogen fixation. Atmospheric nitrogen fixation. Atmospheric nitrogen fixation. The atmospheric nitrogen fixation mean in the thundering and lightning. ඒ කියන්නේ අකුණු ගහද්දී in the thundering nitrogen breakdown and fixing in the thundering, right? In the thundering. No microorganism මේක පාඩමට අදාළ නැහැ, but in next lesson we need to discuss. එතකොට මේක අපි එකට ලියලා තිබ්බම ලේසි, right? In the atmospheric nitrogen fixing mean in the thundering process. Thundering nitrogen breakdown and they are fixing in the soil as ammonia. මේකෙත් මොනවද හැදෙන්නේ? Finally it is producing ammonia, NH4 ions. NH4 ions. There is another process industrial nitrogen fixation මේකත් අර පාඩමේ තියෙන්නේ industrial nitrogen fixation no microorganism involvement, it's a chemical process, but nitrogen fixation, right? Nitrogen fixation. Okay, industrial nitrogen fixation mean inside chemical industries, it's produce nitrogen gas into ammonium fertilizer in the fertilizer production. Again, artificial fertilizer production fertilizer production fertilizer production only what happened, nitrogen artificially it's converted to ammonium ions. එතකොට අපි this fertilizer when we add into soil, it's already release ammonium ion to soil. Okay, they are the nitrogen fixation. මොනවද කරේ, nitrogen gas converted into ammonium ion. Are you clear? The first step done. Okay මෙන්න මේ main first step මෙයා හරිද? මේ වැඩේ තමයි වෙන හතරක් තියෙන්නේ. Okay Tanya? Now the nitrification first step done. ඒව වෙන හතරක් තියෙනවා. There are 4 main ways to convert nitrogen gas into ammonium ions. You know, clear? Okay, if you clearly today I explained things. If you clearly if you can make a small short note including all steps, මේ විදියටම short note එකක් හදාගන්න පුළුවන් නම් definitely you can score full marks for your first structured essay question. Then making very low marks, that's why we doing as the final lesson making podi marks ගන්නක් ඉන්නේ 3 MCQ in first part and part of structured design, right? But lot of theories. But if you can make a short note including each step I explained, definitely you can score full marks. It's a short note එක හරියටම හදන්න. හරි. Now the first step is done. Nitrogen gas converted to ammonia. That's ammonification or nitrogen fixation. Nitrogen fix as an ammonium. Let's move into second main point. In the second main step, what happened? In the second main step, now this produce ammonium ions. මේ එක එක විදියට හැදුණු ammonium ions. Any method produce ammonium ion. The soil contained huge amount of ammonium ion. These ammonium ions using nitrosomonas, special bacteria. Nitrosomonas, very important. Nitrosomonas. This name very important. Nitrosomonas. Special bacteria. This ammonium ion directly convert into nitrite. NO2 minus. එයාට අපි කියන්නේ කොහොමද? nitrite ion. Nitrite ion. Nitrite ion NO2 minus. Still not water soluble. Still not water soluble. This is a final step. Third main step. In the third main step, what happened? It is converted into NO2 minus. Then hadala තියෙන NO2 minus. It is converted into NO3. NO3 mean NO3 mean nitrate ions using nitrobacter bacteria. Nitrobacter. Nitrobacter. Nitrobacter is a bacteria. Nitrobacter is a bacteria. මේ අයන් එක මොකද්ද? Nitrate. Nitrate. This is nitrate. This is the plant available. මම මෙතනින් දාන්න plant එකක්. This is a plant available nitrogen type. ඒව plant එකට absorb කරන්න පුළුවන්. Plant එකට absorb කරන්න පුළුවන්. Okay, this is a full nitrification. If you want to make a screenshot or please note down, this is the full nitrification. Mainly 3 steps, no? Ammonification, nitrite production, nitrate production. Sarmya? Understood? In BST also you're doing nitrogen cycle, no? Am I correct? Tinada? Tanya? BST, I think you have. Dihini? Do you have in BST? The cycles, nutrition cycles? In agri we have. It is same, no? Tanya, it is same mecamethine. Okay. Hari. Okay, complete. Dihini? Complete. Janul? I got full idea. Okay, just remember as a story. First, production of ammonia, ammonia production 4 methods, then produce ammonia, ammonium convert into nitrite first, nitrosomonas, then nitrite convert into nitrate by nitrobacter. This nitrate ion is a plant available, plant absorbable nitrogen type. Okay, can I erase? Take a shot, screenshot. Screenshot tag. Hari. Now, that is nitrification. This is nitrification. Then I told there is another one. This produce nitrogen. This produce nitrogen in anaerobic condition again. Nitrate produce nitrate NO3 minus in anaerobic condition again convert to nitrogen gas. Anaerobic condition. Then anaerobic microorganisms involved for this nitrification process. Anaerobic microorganism involved for this denitrification process. Denitrification process. Okay, who are the anaerobic microorganisms? First one, pseudomonas. 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 Micrococcus. Micrococcus. Pseudomonas, Micrococcus, and Theobacillus. Theobacillus or Thiobacillus dekama කියනවා. Theobacillus. Theobacillus. Okay. මෙන්න මෙයාලා මොකද කරන්නේ? In anaerobic conditions, directly these nitrate ions convert to nitrogen gas. Okay, that we called as denitrification. That we called as denitrification. 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 That's a full story. That's a full story. Okay, who are the denitrification bacteria? Micrococcus, Theobacillus, Pseudomonas. Micrococcus, Theobacillus, Pseudomonas. Micrococcus, Theobacillus, Micrococcus, Theobacillus, Pseudomonas. Pseudomonas, Micrococcus, Theobacillus. Micrococcus, Theobacillus, Pseudomonas. Micrococcus, Theobacillus, Pseudomonas. Hari. Let's move into next part. Do you need break? I can give you 10-minute break. Next we move into back microorganism related industrial purpose that we can discuss within 1.5 hours. That's the end of microorganism. Then evening we discussing plant parts and forest and animals. The lesson is completed. Okay, do you need break? Do you need break? Now 11:20 we can start 11:30. Salmia? Okay. Okay, take a break. Don't leave. Take a break and I'm going to start 11:30, right? 11:30. Microbial production. That's the most important part. Now okay, you can hear, right? Yeah, now you can hear. Good. Hi. Okay, we discussed how to use microorganisms in agriculture. Agriculture mainly involved for soil fertility to maintain cyclic of matters, cyclic of nutrients, very important, nitrogen and other cyclic improvements to keep the balance of each cycle of nutrients. It is very important microorganism, right? Microorganism, different types microorganism, ammonification microorganisms, freely living microorganisms. What are the freely living microorganisms? Acetobacter, right? Not acetobacter, acetobacter, clostridium, and cyanobacteria. Symbiotic microorganisms, nitrogen fixing, rhizobium and anabina. Matakai? Rhizobium and anabina. Rhizobium and anabina. Okay, who is doing this nitrification process? Nitrosomonas, and nitrosomonas ammonium ion convert into nitrite. Nitrite convert into nitrate by nitrobacter. Nitrobacter. Do you remember this nitrosomonas and nitrobacter? Which nutritional group? Which nutritional group? Nitrosomonas and nitrobacter. Nutrition type. What's the nutrition type? Nitrosomonas and nitrobacter. What is the nutrition type? Yeah. What? Chemoautotrophs. Chemoautotrophs. Nitrosomonas and nitrobacter. That's a special group. Special group. Nitrification bacteria nutrition type is chemoautotrophs. Remember that. Chemoautotroph. Hari. Yeah. Utilization microorganism for technical productions, technological productions. Microorganism act as a tiny chemical factors converting raw materials into product. That's a actual advantage. We don't need energy, right? They all have energy, their own energy. Raw material after microbial activity, it is converted into valuable product. Valuable product. Valuable product without any energy or no any conditions requirement, right? We don't need to maintain temperature. They already maintaining the temperature, right? If we provide only favorable conditions, microorganisms doing their job. They are required moisture level, required temperature, and required relative humidity. If we provide optimum condition for microorganism, they doing their own job. Okay, then they convert into raw material into whatever raw material, they convert into valuable product. Valuable product. Right. Right. මේ ටික ඕනේ නැහැ. අපි යන් කෙලින්ම production වලට making of breads. In bakery production, how we use, in food production, how we use microorganism. The first one, bread production, the bread making. Okay, do you know what are the raw materials for bread production? Wheat flour. Yes, wheat flour. Okay, this wheat flour contain a special characteristic. It has a gluten protein. Gluten protein. The wheat flour main nutrition is carbohydrate, correct, but it's contain gluten protein. Gluten protein. This gluten protein giving this the starch, the two particles stick each other. This sticking characteristic given by sticking. Stick වෙලා තියෙන්නේ. ඇලෙන වගේ characteristics, right? The sticky characteristic given by this gluten protein. right? It's a special type of flow, right? Now, give me a second. Sorry. Okay, I told you the wheat flour are the main raw material for bread production. This wheat flour contain the main protein is gluten protein. Please note down that it contain gluten protein. This gluten protein giving this characteristic sticky character given by the gluten protein, right? ඒක නිසා තමයි අපිට පාප් විදිහට use කරන්න පුළුවන්. It's contain sticky characteristics. හරි. බලන්න. Now what is the first step of bread production? We have to add water and make as a pulp. Make as a pulp. Make as a pulp. Sorry. Before make as a pulp, we have to add Saccharomyces cerevisiae. Saccharomyces cerevisiae, that means yeast. 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 Right? We have to add some very small amount of baking powder. Baking powder means yeast. Baking powder. It's a fungi, Saccharomyces cerevisiae. Saccharomyces cerevisiae. And if you want, we can add small amount of sugar. Small amount of sugar, right? Sugar, small amount of sugar, and add yeast and add water and make as a pulp. Make as a pulp, right? Make as a pulp. Hard. Now we have to close this container and keep some time for the fermentation process. Fermentation process. You know, yeast means Saccharomyces cerevisiae. It's facultative anaerobic. Therefore, if we give anaerobic condition, like a less oxygen environment, it is favorable for Saccharomyces cerevisiae. Oxygen, normal environment also okay, but if we provide little bit anaerobic, less oxygen environment, it is more favorable because the Saccharomyces cerevisiae is a microorganism which group? Facultative anaerobic. Anaerobic එකක් දුන්නොත් වැඩි හොඳයි, but Arabic environment also okay, right? Right. Now keep some hours. Keep some. Normally we have to keep 3 to 4 hours. 4 hours. Then what happened? This wheat flour also contains some amount of sucrose, sugar amount. If not, we can add additionally 1 spoon of sugar. Podi amount එකක් දාන්න. හරි. Not for taste. It's only for microorganism substrate. Microorganism substrate. Sugar adding as a microorganism substrate. Okay, then what happened? What happened? This sugar mean sucrose. This sucrose break down. These Saccharomyces cerevisiae enzyme release invertase enzyme. Remember, invertase enzyme. Invertase enzyme and break down this sucrose into glucose and fructose. Glucose and fructose. Sucrose break down into glucose and fructose. This glucose, now it is produced glucose. Now glucose means C6H12O6. This is a glucose. The Saccharomyces cerevisiae. Saccharomyces cerevisiae. Saccharomyces cerevisiae. Saccharomyces cerevisiae work on this glucose. This අපි add කරපු සහ wheat flour වල තියෙන glucose fermented by Saccharomyces cerevisiae. Then what are the product? It producing C2H5OH. What are them? C2H5OH. What is this, Charol? C2H5OH mean ethanol. Ethanol plus carbon dioxide. It's produced ethanol plus carbon dioxide. It's ethanol, right? Ethanol. Ethanol. Ethanol plus carbon dioxide. Ethanol and carbon dioxide. Then what happened? They produce ethanol deposit in holes and carbon dioxide due to the sticky characteristic of this mixture, this pulp, this carbon dioxide is fill inside the holes. මේ වතර fill වෙනවා. Right? Due to the sticky characteristic මේක fill මේ එළියට යන්න බෑ. Some part of carbon dioxide releasing, but lot of carbon dioxide fill inside holes in the mixture. Understood? Due to that, we call the mixture is increasing in volume, right? Why? The carbon dioxide is fill inside the holes. I got carbon dioxide fixing in the holes, right? This producing carbon dioxide. I got. That's why we called අපි අර පිටි එක පිපෙනවා කියලා කියන්නේ. Now the starch volume is increasing. Okay, that's the bread production. Now we can get after 4 hours we can get this pulp, starch pulp, and cut into small parts, same size parts, and put into molds. අපි molds වලට දානවා. මේවා molds වලට දානවා. Bread production molds වලට දානවා. මේවා දානවා molds වලට, right? Then place in a oven. Place in the oven. In the oven, what happened when it is heated? When it is heated into higher temperature, this ethanol evaporate. Why? Ethanol is highly flammable and it is very low boiling point. It's easily evaporate at very low temperature. It's අපි heat කරද්දි මෙයා evaporate වෙනවා. But this carbon dioxide fixing inside the holes, what happened? They evaporate. Gas evaporate when heated. That's why the soft holes made in the bread due to this carbon dioxide evaporation. Carbon dioxide evaporated atama. There was an exam question: what is the text? How to form the texture of bread? A texture එක හැදෙන්නේ කොහොමද කියලා අහලා තිබ්බා. Then what is the reason? In the heat, in the Saccharomyces cerevisiae produce ethanol and carbon dioxide. This carbon dioxide is fixing in the holes. In the heating process, this carbon dioxide evaporate. එයාලගේ volume එක වැඩි වෙනවා, evaporate වෙනවා. Not evaporate, expand වෙනවා. Evaporate වෙන්නේ ethanol. Carbon dioxide, as a result of expansion of this carbon dioxide, the volume of mixture increasing and the texture getting very soft texture. Not like pulp, it's getting very soft texture of the bread. Understood? This is the bread production. Very simple. Any doubt? Any doubt? Very good. Okay, what happened? First add yeast and small amount of sugar and mix the wheat flour and add water and make as a pulp. Then this pulp contain small amount of sucrose. This Saccharomyces cerevisiae work on this sucrose and convert into glucose and fructose. This producing glucose again fermented by Saccharomyces cerevisiae. It's produce ethanol and carbon dioxide. This ethanol and carbon dioxide fixing inside these holes of the pulp. Right, that's why the volume of mixture is increasing. In the heating process, now this pulp cut into small pieces and put into molds and placed in the oven. In the heating process, ethanol evaporating, carbon dioxide expanding. That's why the soft layer, soft texture of the bread is formed. Okay, Tanya, clear? Tanya? Okay, that's the microbial usage. Microbial usage. Please, if you want, please note down a short note. Take a short note. Can all clear? Udanya? Fatima? Understood? Kabir? Remember step by step. What's the first step? Get wheat flour and add sugar and yeast and water. Make as a pulp. In the fermentation process, sucrose break down into glucose, and glucose fermentate by Saccharomyces cerevisiae, producing of ethanol plus carbon dioxide. In the heating process, this ethanol vaporize and carbon dioxide expand. Due to this expand, now think this is a mixture. It's contain carbon dioxide like this. Carbon dioxide, small holes filled with carbon dioxide, right? Now what happened in the heating process? This carbon dioxide expanding, expanding like this. That's why it's forming holes inside breads. As a result, it's forming this soft textile, soft texture of the bread is forming due to this carbon dioxide expansion. Carbon dioxide expansion. Carbon dioxide expansion. Ethanol, what happened? Evaporate in the heating process. Right? Udanya? Clear, right? Sarmya? Tanya? That's the bread production. That's the bread production. Okay, let's move into next one. Okay, මේ දීලා තියෙන process එක. Air holes of carbon dioxide gas bubbles through the flow of mixture. Happens ethanol seasoned evaporate in the baking process. Okay, this amino acid production is not very important. Normally microorganisms use for different amino acid production. Sometimes essential amino acid production. Essential amino acid means amino acid that we can't produce in our body. Essential, as example, lysine and methionine. They are essential amino acids. Essential amino acids. That's a production of glutamic acid. Glutamic acid also type of amino acid. That one also essential one. That's producing by bacteria Corynebacterium glutam. Corynebacterium glutamicum. Yeah, that contain glutamic name. Corynebacterium glutamic. Corynebacterium glutamic. For amino acid production, we use genetic engineering technology. I will explain in the middle part genetic engineering technology. Now remember, for glutamic acid production, Corynebacterium glutamicum is the bacteria. Corynebacterium glutamicum is the bacteria. Corynebacterium glutamicum is the bacteria. Corynebacterium glutamicum. Okay, lysine production. Lysine production, remember the same bacteria. Make a history Corynebacterium glutamicum, different strain. ATCC1327 strain used for lysine production. Maybe the production I'll explain in the next genetic engineering part. Alcohol beverage production, the most important part. Alcohol beverage production. Make it very simple. Try to get a short note, right? Try to get as a short note. Alcohol beverage production. Our first alcohol beverage is 2D production. Any alcohol production, we use Saccharomyces cerevisiae types. Saccharomyces cerevisiae means yeast. Okay, what happened? Any yeast glucose C6H12 O6 fermented by Saccharomyces cerevisiae, what happened? It's producing C2H5OH. It is ethanol. This is a bread had in very small amount. Now we doing this process in purposely produce of ethanol. Now glucose solution fermented by Saccharomyces cerevisiae. Then it is produced ethanol plus carbon dioxide. Now this is ethanol. Carbon dioxide is releasing. Okay, this is a simple ethanol production process. Hari. Make it very simple. The 2D production, you know what is the raw material for 2D production? Quickly, Kabir. What's the 2D production raw material? What's the raw material for 2D production? 2D mean monad. 2D can happen. Kenul, what are the meaning of 2D? Oh, Ra kila kiyan? Hitul Ra, Pol Ra, coconut 2D. Hitul 2D. Yeah. 2D, Ra. The main raw material, the sucrose solution extracted from the inflorescence of Kitul coconut, this type flower inflorescence extract sucrose solution like this. This solution is the main raw material. Sometimes if not, we can use sugar solution also. Okay. It is glass of water and add water, right? Add 3 to 4 spoon of sugar, right? Right? Now keep it in open environment. Okay, after 3 to 4 days, what happened? It's converted to D. No need to add yeast because in our environment, lot of Saccharomyces varieties. Saccharomyces cerevisiae, when we provide a nutrition media, environment available microorganisms growing inside our solution, right? Then they growing rapidly and convert this glucose into 2D. 2D mean glucose into ethanol plus make it in sucrose ticker. The contain sucrose. Now it is seen in them it's sucrose converted into glucose by invertase enzyme. This glucose means C06 by Saccharomyces cerevisiae, this environment available Saccharomyces cerevisiae convert into C2H5OH plus carbon dioxide. And the water amount is also there. Water soluble ethanol mixture is water soluble ethanol mixture. වතුරේ තියෙන ethanol ටිකක් අපිට හම්බෙන්නේ. Normally 2D, how about ethanol percentage? How about ethanol percentage of 2D? Kabir, what do you think? 2D ethanol percentage? තමනේ කොච්චර වගේ percentage එකක් තියෙනවා? You know, how about ethanol percentage of 2D? Tanya, ethanol percentage of 2D. රාවල තියෙන ethanol percentage, yes. Yeah, 6 to 7, some 5 to 7 range. 5 to 7 range. The ethanol percentage depend on the sucrose solution. It can glucose තියෙන amount එක අපේ solution එකේ. If we add more sugar, we can increase ethanol percentage. We can increase ethanol percentage, right? Okay. Now for natural make up සීනි දාලානේ හැදුවේ. For natural 2D production, we get the sucrose solution extraction from the ඒකට අපි කියන මීරා කියලා. Extracted from the inflorescence of coconut ketol flowers. Okay. Okay, normally this is just sweet taste solution, right? Sweet taste solution make no ethanol. This extraction no ethanol, right? ඒක බිවට මුකුත් වෙන්නේ නැහැ. It's just king coconut water, right? If you want, we can use coconut water as well. Coconut water also we can use for this 2D production, because that's contain also some sucrose. If you can add okay, right? Hari. I told you in 2D production very simple. May marley scene. If you want, make note down, right? Collect the sucrose solution from the inflorescence of coconut or ketol flower, right? Add Saccharomyces cerevisiae, that means yeast, small sample of yeast, and keeps of fermentation. Then what happened? It is converted to ethanol and carbon dioxide. The sucrose contained in the solution converted to ethanol and carbon dioxide. This ethanol is dissolved in the solution water. It contains some water amount. ඒකේ වතුර වල දිය වෙනවා. Carbon dioxide is released. Carbon dioxide releasing, then we can get ethanol solution mixed with water. This water mixed ethanol solution is the 2D. Normally 5%, normally හදන එකේ 5 to 6% of ethanol and the 94% of water. 94% of water. Okay. Clear? 2D production. 2D production. Harned. Me Sharon? Understood, right? Quickly. Then make up it our product in up to the 1 year part, me 1 hour part. Let's try to complete as soon as possible. It's very simple. Me okay, one by one production samadhi. Now the difficult part actually we already discussed. The difficult part microbial classifications, nutritional method, respiration methods. The next one, wine production. Okay, what is the main raw materials for wine? What's the main raw materials for wine? Wine. What's the main raw materials for wine production? Dihini. What's the main raw materials for wine production? Yeah, grapes. Grapes are the raw materials. Grapes are the raw materials. Okay, if you want this characteristic taste, we have to use grapes. The other fruits also use karanna puluwang, but if you need this characteristic taste, we have to use grapes. Grapes. See, the meaning of the word of wine is clear that it's drink produced from grapefruit. Wine kiyanne මෙත්තනම grapes වලින් හදන එක තමයි. Right? But wine can also produce by using fruit juice as well as king coconut water as well. But fruit juice and king coconut water with sugar content of about 10 to 25 are more suitable for wine production. The main sugar in juice are glucose, yes, fructose and sucrose. Fruit juice with high acidity are ideal for wine production. If it is high acidity, because this is microorganism. For wine production, we not going to use simple normal yeast. We use special type of yeast. I will explain what is the type. Special type of Saccharomyces species that's already cultured in the laboratory. Especially had the microorganism use kara. Okay, the first thing, the wine production, we can classify it into four main states. The first one, the preparation of juice. The second one, fermentation. Third one, clarification. The final one is AG. Apni ළඟට තියෙන beer, right? කොම alcohol හදන විදිය දැනගන්න පුළුවන්. හරි. හරි. බලමු. Okay. Now the first step, the preparation of fruit juice. Fruit juice mean we can use red grapes, not red grapes, black grapes, black grapes and yellow green, one of the red skin and green. Yeah, both grapes we can use, but the most suitable one is red one. Red mean dark color one, black grapes. Okay, first thing, before that, do you know this is a grape. අපිට මේක red එකේ නම් හොඳටම බලන්න පුළුවන්. Think it is a grape. Grapes, the surface of the skin. Do you have seen some wait? Do you have seen some fungi attraction? Make it just white color spot. Do you have seen white color spot on the grapefruit on the skin of the fruit? Okay, what are they? They are a fungi type. Grape skin it's a good media for fungi development. fungi development, right? It's a type of fungi. But due to this skin, they can't directly attack to this the internal fruit. If there any damage, rapidly they attack to this fruit. Again, make a පොඩ්ඩක් හරි damage වුණොත් skin එක ඉක්මනට නරක වෙනවා. The reason the fungi develop on the skin, right? Normally environment available fungi develop on the skin of the waves because it's a good media for them. Good media. Right? I think good. Okay, now the first thing, the first thing, our first step of vape is make as a pulp. Then what's the meaning of pulp preparation? The pulp preparation mean what? Yeah, the pulp preparation mean these grapes with skins. අපි ඉස්සෙල්ලාම කරන්නේ wash කරලා just crushing, right? Crushing and convert into, see, for red wine production, red skin or non-skin grapes or colorless grapes, that means green one, are crushed and make the juice as a juice, right? Make as a juice. Okay, then what happened? It's just a mixture of fruit parts and fruits again, grape parts and mixture, right? Juice. It's a mixture of juice. Okay, without filtering. තාම අපි filter කරන්නේ නැහැ. කියන්නේ මේ juice it's a juice, fruit juice with grape parts. No problem, right? අපි chop කරලා convert into juice without filtering. තාම filter කරන්නේ නැහැ. හරිද? Then inside this type container, add the fermentation mean specially introduced, specially cultured microorganism before fermentation. Now I already explained this white color spots. This white color spots are fungi. When it is crushed, අපි wash කරාට ඒව යන්නේ නැහැ. Wash කරන්න බෑනේ අපිට soap වගේ use කරලා because it is fruit නේ. Then if we wash, only dust particles removing, but these fungi spots remain, right? When it is crushed and convert into pulp, then what happened? These fungi enter to the juice. Then what happened? If it is fermented, if it is go to fermentation process directly, this fungi attack to the fermentation bacteria. Not bacteria, fungi may vulnerable. Vulnerable microorganisms. Environment microorganism affect to the cultured microorganism. It will make it, we can't get a specific taste. We can't get, right? It's producing different product with different microorganisms, right? Due to that, the first step, we have to pasteurize this mixture. What's the meaning of pasteurization? These already available microorganisms need to be destroyed. For this process, we have different process. These microbes include bacteria, yeast, and other fungi. Wine can be made by allowing natural fermentation. මෙහෙම natural fermentation එකකින් හදන්න පුළුවන්. But the problem is commercial wine production, natural microbial population is removed either by applying sulfur dioxide or by sulfur dioxide අපි මේකට this juice flowing through sulfur dioxide mixture. Then what happens? Sulfur dioxide is a pasteurizing gas. Due to the sulfur dioxide, the sulfur dioxide is toxic to microorganism. Then it will be pasteurization. After pasteurization, අපි කොහොමද pasteurize කරන්නේ? Using sulfur dioxide and pasteurization. Then already available microorganism in the juice we can destroy, right? 99% we can destroy. We can't 100% not possible. 99% we can destroy, right? After that, especially and high quality cultured Saccharomyces carsongensis. No, Saccharomyces cerevisiae strain. Saccharomyces carsonensis for beer production. Sorry, Saccharomyces strain. Saccharomyces cerevisiae specially cultured strain, no specific name, enter to the add into the mixture and keep some days, few days, 1 to 2 days for fermentation process. Fermentation process. This fermentation process we called as incubation. Incubation. Do you know what's the meaning of incubation? Incubation. අපි මේ eggs වලටත් කියන්නේ incubate කරනවා කියලා නේද? eggs විතර රකින්නවට කියන්නේ incubate කරනවා කියලා නේද? Yeah, incubation mean provide suitable conditions, temperature, relative humidity, moisture, provide suitable condition for microorganism development. That's called as incubation. Okay, now hari api I බලමු first may from first point. First get the red or green grapes and chop and convert to pulp and juice, fruit juice, grape juice, and it contains lot of environment microorganisms. The second step, pasteurization. What we use for pasteurization? Sulfur dioxide gas. After that, specially cultured microorganism Saccharomyces strain introduced to the mixture, then keeps favorable condition for fermentation, that we called as incubation. That's we called as incubation. That's we called as incubation. That's we called as incubation. Okay. Up to now, okay. Up to now, okay, right? Okay. After incubation, what we doing? Clarification. Clarification means just filtering. After fermentation is complete, now we can filter using filters. Separate solid particles. Then solid particles තිබ්බනේ මේ grape particles. After fermentation, filtering. Hari api what are the steps? Preparation of pulp, pasteurization, fermentation take place, clarification mean filtering. After that, we have to fill into this type barrels. Well, දැකලා තියෙනවද මේ wine barrels? කොහෙද මේ වල දැකලා තියෙන්නේ? These wine barrels? cartoon එකේ තියෙන කොහෙද? Do you have seen these barrels? බලලා තියෙනවද සූරපප්? Yes. They are wooden barrels, specially prepared wooden barrel made by woods, right? Wooden barrels. We have to fill into these already produced wines, fill into wooden barrels and keep 6 months, 1 year. That's for aging. Actually, when it is filtered, after filtering process, it don't have specific required taste or smell. To get this the wine specific taste and color and everything, all characteristic, we have to keep some time for aging. Again, pulp වෙන්න තියන්න ඕනේ නැත්තන. The value of this wine is depend on the aging period. Sometimes new wine is very low price, but old wine are more expensive because the aging period very important. They complete the fermentation process and improve wine's quality and aroma due to this aging period. Hari. Thank you. What are the steps of wine production? First one, grape pulp preparation. Second one, pasteurization by sulfur dioxide gas. Third one, fermentation by specific microorganism Saccharomyces strain. Filtration, clarification, and aging. Store in wooden barrel and keep long time. මේක තියෙන time එක depend වෙනවා, right? Sometimes we use මේ 9 months, 6 months, 1 year තියෙන ඒවා තියෙනවා, right? More than 1 year wine also there. In doubt of wine production? Sharon? Jenol? Please make a short note. What's the first step? Grape pulp preparation. Second one, make a more lazy. The thing is no essay questions, no. It's going to make a fully you can go with short note because you have 4 MCQ questions and 1 part of structured disease. I'll make a full short note. Give me a second. Okay. Are you clear? Steps of wine production. What are the steps? Grape pulp preparation. Filtration after that's the point. Right? The filtration coming after fermentation. එතකම් filter කරන්නේ නැහැ. ඒ grape particles එක්කම තමයි ferment කරන්නේ. Right? Grape pulp preparation, that's a juice. Then pasteurization by sulfur dioxide gas, make a short note. And fermentation by specially Saccharomyces strain. Clarification, that means filter solid part, filter out solid part, and aging for longer time in wooden barrels. Okay, okay, Fatima, understood? Kabir, quickly. Tanya. The wine production is done. Let's move into beer production. Do you know what are the raw materials for beer production? Beer හදන්නේ මොනවද දන්නවද? Beer. Wine grapes, beer production. Beer producing by me. We can use any starch based we can use rice as well. Rice, maize, and wheat also. Wheat. But the problem, they are expensive. Therefore, in the world, mainly using barley. Barley. Do you know what are the barley? Barley is a type, seed type, like maize. Barley. Rice also okay, but it is expensive. But barley cheap price, and it's easily growing and high harvest. Due to that, barley is the main product, main seed used for beer production. Okay, let's see what are the steps of beer production. The first step make an mehem. The first one, malting, very important. The first one, malting. The second one, mashing, malting, mashing. They are very simple process. Early fermentation. Next one, fermentation, and after fermentation, no aging period for beer. Immediately we can use. Immediately we can turn into can and fill into can and send to market. What are the steps of beer production? Okay, what is the main raw material? Barley seeds. Okay, normally what is the barley seeds? What is the main ingredient contain? What's the main carbohydrate? Starch. Starch contain, now seed, it's contain starch. But is it ferment by Saccharomyces cerevisiae starch? No, starch is a polysaccharide, not ferment by Saccharomyces cerevisiae. The first step, the malting, mean we need to wet this barley seed in water and keep some time for germination, seed germination. In the germination process, this it's producing amylase enzyme. It's producing amylase and maltase enzyme. What happened? This starch convert into glucose. I got the first step, malting. Malting is the first step beer production. Malting. The first step, malting. Please take a short note. Malting. In the malting process, what happened? Malting will be in the malting process, the barley seed contain starch convert into amylase enzyme, then it is converted into glucose, breakdown into glucose. Okay, this is the malting. Very simple. The starch contained in barley seed convert into glucose in the germination process. In the germination process, amylase enzyme break down this complex starch into simple glucose. Right? The first one is done. First step is done. Okay, that's the first step. We then germinated barley seeds. See, germinated barley seed. Okay, the second one, mashing mean simple. It's grinding. Grinding mean these barley seeds, these convert into powder. Grind karna, right? Mashing. The second step, mashing. Grind into powder. Powder form. Grind into powder form. Grind into powder form. Then if there are any in the mashing process, in the grinding process, if there are any unreacted starch or maltose complex break down glucose. That one also break down into glucose, and the final product, this final powder mixture, we called as the malt. Malt. The final product we called as the malt. Methanic passe make it malt, right? Malt. Malt is the name. Malt. Okay, the third step is the early fermentation. First one, what is the first step? Malting. The second one, mashing, grind into powder. Then early fermentation. Early fermentation, very, very important. But in the early fermentation process, we have to add, do you know me, what is the taste of beer? Then the beer taste එක බීලා තියෙන්න ඕනේ නැහැ. දන්නවද මොන ටේස්ට් එකක්ද තියෙන්නේ? චරෝ you know, do you know? What is the taste of beer? Normally, the beer, no sweet, no sweet. පැන් රස නැහැ. පැන් රස නැහැ. බලන්න. Others, you know, not sweet. Kabir, any idea? What's the taste of beer? Normally, it's තිත්ත රසක් තියෙන. Bitter taste. Bitter taste. තිත්තයි. ඒ කියන්නේ the bitter taste with some spicy, right? Okay, then to get this characterized bitter taste, now the sugary solution formed by the enzyme in malt is called as wort. Initially this powder is malt, now it is called as wort කියලා, right? Wort මේක එක එක නම් තියෙනවා, හරිද? Wort. Now in the early fermentation stage, it is wort. Wort is the name. The solids in this sugar solution are removed and dry beer flowers. Beer flowers mean there is a plant, Homulus lupulus. This Homulus lupulus that can be beer plant එක කියලා. This Homulus lupulus plant flower inflorescence contain flower inflorescence contain flower එක inflorescence එක dry කරාම that we call as hops. What දෙක තියෙන්නේ? Hops. Maybe hops. Simple, right? Now the malt mixture, now we called as wort. Into this wort mixture, we have to add dry Humulus lupulus. That means beer flower inflorescence එක දාන්න ඕන. Right? That we call as hops. Right? That we call as hops. These hops mean the characteristic bitter taste coming due to this hops. අපි hops දාන නිසා තමයි ඒක හැදෙන්නේ Humulus lupulus plant එකේ. Right? Right. හරි. Then this hops stop any further enzyme activity, no any enzyme activity, and adds a distinctive flavor for the beer. That means bitter taste given by this hops, and it's avoid the enzymatic activity again. Early fermentation stage into what solution at hops where we collect hops from beer flower. Beer flower means Homulus lipulus flower, right? Homulus lipulus flower. And now fermentation. In the fermentation process, we have to introduce special cultured microorganism that we call as Saccharomyces casparensis. Saccharomyces casparensis type, and incubate for control condition. After fermentation complete, we can fill into can and immediately send it to market. Okay, let's revise beer production steps. First one, germination of barley seeds, that's malting. In germination process, starch convert into glucose. The second step, malt mashing, convert into powder, convert into grind and convert into powder. Early fermentation, add hops dana the Humulus libulus plant. Hops at beer flower add karna to stop further enzyme activities and to get the characteristic taste, bitter taste. And fermentation, and add water and make a solution එකක් විදිහට හැදුවම තමයි what කියන්නේ, sorry. Early fermentation. Third one, early fermentation. Make solution and add hops done. Add hops. Now that we call as what can that? Okay, step by step, name is different. Now it is what? What sorry. What can mean? What? Malt, now what? Right? Now in the fermentation process, fermentation process at Saccharomyces casparensis. Saccharomyces casparensis for fermentation, then incubate period, then fill into cans and send it to market. No aging period only for wine. Aging period only for wine. Aging period for wine. Hari, okay, today we can complete. Before 1 PM we can complete. Very simple thing. Hari. Evening we can complete this lesson, right? Don't miss anyone, right? Evening class. Okay, that's a beer production. Okay, let's revise again. First, malting, barley seed germination. In the germination process, complex starch by amylase enzyme convert into glucose. Mashing, germinated seed grind and convert into powder, that's malt. In the early fermentation process, add hops, that humulus libulus plant flower inflorescence, then add hops, then stop further enzymatic activity and giving bitter taste, characteristic taste, and add Saccharomyces carsongensis, especially for cultured microorganism type, fungi type, and fermentation. Remember, Sharon, Tanya, Sarmya, Sabir, Yanul, clear? Now you have to remember that මේ short note එක විදියට මතක තියාගන්න. Step by step that මේව explain කරන්නේ නැහැ. Because now I say questions. Yanul, clear, right? Fatima, understood? Right. Okay, let's move into final. Vinegar production. Vinegar production also very simple. Vinegar production mainly two types. Vinegar production, ඒකත් හදන්න short note එකක්. Vinegar production. Vinegar production. Vinegar production mainly two steps. The first step you already know. First step mean C6H12O6, any sucrose solution fermented by yeast and convert into C2H5OH and carbon dioxide. Convert it to ethanol solution. It can a production of 2D. 2D වලින් තමයි 2D is the main raw material for vinegar production. අපි 2D හදලා ඉන්න ඕනේ. That's the first step. That's the first step. Any sucrose solution convert into ethanol mixture, carbon dioxide releasing. Then this ethanol, then ethanol මම මේක අද ඔයාලට ගෙදරදී හදලා බලන්න කියලා දෙන්න. හරිද? Send me photo after within 1 week you can do this experiment. Right. Now you got sucrose sample. Sucrose means you got glass of water and add 4 spoon of sugar and keep 4 days. If you have, you can add yeast පොඩ්ඩකුත් දාන්න පුළුවන්. එතකොට වැඩේ ඉක්මන්. Right? Within 2 days you can get 2D if you add yeast. Right. Now this C2H5OH, that means what? C2H5OH means ethanol. C2H5OH, ethanol react with oxygen. Ethanol react with oxygen mean we have we have to introduce special microorganism type that we called as Acetobacter acidic. Acetobacter acidic. Simple, acidic. Acetobacter acidic. And gluconobacter. Gluconobacter. Acetobacter acidic and gluconobacter. Gluconobacter. Acetobacter acidic and gluconobacter. Using this Acetobacter acidic and gluconobacter. Acetobacter means Arabic, right? I told you. Acetobacter acidic and gluconobacter bacteria, what they're doing, this ethanol oxidize and convert into CH3COOH. CH3COOH. This is acetic acid. Acetic acid. Acetic acid. Acetic acid. Plus water. This water and acetic acid mixture we called as vinegar. Vinegar. Water and acetic acid mixture we called as vinegar. Water and acetic acid mixture we called as vinegar. හරි. මෙතන ලියන්න තියෙන්නේ tubes නැතිව මේක ලියාගත්තත් ඇති, right? This is the second step. Tubes තියෙනයි අර space එකේ ලියන්න. If you have tube, note down the space. If not, the short note also enough. But you need to remember the vinegar production mainly two types, two steps. The first one, glucose convert into ethanol by Saccharomyces cerevisiae, that you already know. This produced ethanol mixture react with oxygen by Acetobacter acidic and Gluconobacter. Then it is produced acetic acid and water. This acetic acid we called as the acetic acid and water mixture we called as the acetic acid and water mixture is a vinegar. Acetic acid and water mixture we called as a vinegar. Acetic acid and water mixture we called as a vinegar. Hari. Any doubt? Very good, Kabir. Hari. Now we have dairy production and compost and bioremediation. Body part 20 minutes part. Api now tired. Let's do evening. Let's do evening. Dairy production, very simple part. Hari. Now you have now 12:45. Now you have 6 hours. Up 6:30 you have 6 hours. Go through again this theory. Then I'm balanda. You can complete the SFT syllabus, right? Again go to this tube up to vinegar production, and let's start evening from curd production, milk production and compost. Plant product and forest and tissue culture animals. Complete. Okay, do you have any doubt up to now? Khabir, Fatima, Udarna? Udana, you have class evening, right? Okay, no problem. If anyone miss evening class, please watch recording today itself. Adam balanda, adar mahari balanda. Don't keep anything to do next week. To do next week. Okay, go through again and let's meet evening 6:30 p.m. You can. Okay. Let's meet evening. Goodbye.